Hello, everyone, and welcome to our webinar entitled Disrupting Life Science, How VHH Antibodies Will Shape Future Research. I'm Jackie Oberst, Assistant Editor for Custom Publishing at Science, and I'll be the moderator for this discussion. Before we get started, I'd like to orient our online viewers to what's being currently seen. At the top right of your screen, you'll find a photo of today's speakers and a View Bio link, which you can click on to read more details about their background and research. To the right, you'll also see the Resources tab, where you can find additional information about technologies related to today's discussion and a PDF of the slides. You can also log into your Facebook, Twitter, or LinkedIn accounts during the webinar to post updates or send tweets about the event. Just click the relevant icon at the bottom left of the screen. For tweets, you can add the hashtag #ScienceWebinar. During the panel discussion, we will accept and answer some of the viewer questions. So start thinking about some questions now and submit them at any time by clicking the Ask a Question tab, also on the right, typing the question into the message box, and then clicking Submit. Finally, a warm thank you to Jackson Immunoresearch for sponsoring today's event. I'd now like to take the opportunity to welcome a really fantastic panel today. I'll give each of them a chance to say hello and introduce themselves, starting off in the West Coast with Dr. Natalie Castellano.
Thank you so much for the intro. I'm excited to be here for this panel. I'm Natalie Castellano, CEO at Abtera Biosciences, where our mission is to discover the next generation of antibody therapeutics. We've been working with camelids like llamas and alpacas for about the past 5 years, and shameless plug, we're hiring, so across the board, dry lab, wet lab, commercial. If you're interested in trying out biotech or near graduation, we'd love to hear from you. So please do contact me after if you're interested in talking about a position.
Great. Thank you, Natalie. Next, I'd like to head over to the East Coast to welcome Dr. Brian Chait. Welcome, Brian.
Hi. I'm also delighted to take part in this webinar. I'm a professor who works at the Rockefeller University in New York City with a special interest in analytical techniques applied to biology, and with a great current interest in nanobodies and their applications. Thanks.
Thanks, Brian. And finally, back to the West Coast with a warm welcome to Dr. John Aitchison.
Hi, thank you. I'm happy to be here as well. Thank you for the opportunity to join in today. I'm a principal investigator at Seattle Children's Research Institute and professor at the University of Washington in the departments of biochemistry and pediatrics. Specialize in system cell biology and global infectious disease. And recently we've been exploring nanobodies to combat infectious diseases such as SARS-CoV-2.
Okay, thank you, John. The speakers are going to start with a brief introduction of their own. We will then begin with some panel questions, as well as sprinkle in some questions posed by our viewers.
So, by way of introduction, let me say a few words about what nanobodies are and what I consider to be some of their potential advantages over conventional monoclonal antibodies. Conventional antibodies like the IgG shown top left consists of protein chains, too heavy and too light, with a total molecular weight of about 150 kilodaltons. In camelids, which include various types of camels, llamas, alpacas, etc., they have in addition to these conventional antibodies, they have heavy chain only antibodies, like that shown in the middle. Cutting away the bottom conserved part, the so-called FC effector domain, leaves the antigen binding variable domain shown on the top right. This component is termed the VHH fragment or nanobody, with a molecular weight around 1/10 that of a conventional antibody. So it's small, It's small, it lacks the light chain, and it lacks glycan modifications, and there's no Fc effector domain. However, it maintains full antigen binding capacity, and shown in the bottom there, you can see the so-called paratrope, the part on the nanobody or antibody that binds the antigen. And the paratropes on the nanobodies take up a large component of the molecule, and so it is small and can get into places that perhaps steric difficulties with monoclonal antibodies prevent. It's highly stable and very soluble, and can be dried and heated to quite high temperatures. So one can keep this under relatively robust conditions and still maintain full activity. At the same time, the affinities are completely comparable to normal antibodies and can be very high in the nanomole to picomole range. And because they're little and because the paratropes are a large part of the molecule, they have very, very high on rates. So typically they're between 1 and 2 orders of magnitude faster than are the best antibodies. They generally have fairly low immunogenicity and are readily humanized. They can be cloned and expressed in bacteria for easy and affordable production. They're littles, and you can express them in bacteria in little units. So you can multi-target them and make a series of plug-and-play type formats that allow you to tune things like half-life and multiple targets and so on. And finally, they're very robust and small, so you can think about using them in administration to airways, directly to the lungs, perhaps penetrating solid tumors, that sort of thing. To show you the structure of a typical nanobody, in this slide, you see the conserved beta-stranded framework region, and it supports 3 so-called complementary determining regions, so-called CDR loops, that define the epitope binding paratrope on the nanobody. And the largest and perhaps the most important of these is CDR3. And I believe Natalie will say a little more about this next.
Yes, thank you. So at Aptera Biosciences, we are very interested in VHHs and nanobodies, but we also look at other species. So we work also in conventional antibodies from humans and rabbits and mice. And one of the technologies that we use to look at the immune response is next-gen sequencing for immune repertoire analysis. So when we started working in nanobodies, we kind of took a bird's eye view at what the repertoire looks like in llamas versus how it looks in other species. So I just have a couple summary plots of kind of observations that were unexpected. So when we think about the purpose of the adaptive immune system, of course, diversity is a critical part of it. That's how we deal with any pathogen or any, kind of foreign invader in our bodies, is that we're able to generate immense sequence diversity, and so we can get good binders to any of those particular immunogens. But interestingly, for nanobodies, they are constrained to some extent because they don't have the added diversity of the light chain. There's only one polypeptide chain that really can generate diversity and bind to these particular targets. So what was interesting when we started looking at llama repertoires, and this is true across several dozen different llamas, is that it's really dominated by a small number of germline genes that rearrange to produce the VHH sequences. So here what I'm showing is just a histogram. Along the x-axis we have the germline V genes, so both ones that are incorporated into conventional antibodies, but then also ones that are incorporated into VHH antibodies. And unlike other species, we see the VHH repertoire dominated by a very small number of germline genes. So this was surprising to us just because, the nanobody is already handicapped in terms of achieving diverse repertoire because of the lack of light chain, but it's even further hindered by the fact that it's only incorporating a small number of germline V genes. So the nanobodies have to use other mechanisms to basically make up for that lack of diversity. So one way in which we found nanobodies to be unique is that they tend to be actually more mutated than other species. And this is just looking at a non-immunized, quote-unquote naive llama. We're looking at a histogram of mutation load per antibody. And so here on the x-axis is just the count of amino acid mutations from the germline rearrangement and then the frequency of those in the repertoire. And we're looking across 4 different species. And so we see mouse splenocytes actually are really kind of low mutation load. Rabbit here in red is a little bit more mutated. Human PBMCs, we see kind of quite a spread of mutation load, and then llama nanobodies tend to be much more mutated than other species. And again, this is something that we've seen persist across individuals and even in the kind of non-hyperimmunized state. The other way that nanobodies add diversity is through their CDR3. And so we see, in general, VHH repertoire has longer CDR3 sequences than other species. So here I'm just showing a CDR3 length distribution of amino acid length of the CDR3 and then frequency across those same 4 species. So we have mouse in blue trending toward kind of the shortest CDR3s, and then we have rabbit and human in red and green. I apologize to colorblind people, this was a bad color choice. And then we have llama here peeking out on the right with the longest CDR3s on average. And at this point, I'm handing it off to John, who can talk a little bit more about developing nanobodies and their uses.
Hi, thank you. I just want to tell you a little bit about how we might generate nanobodies. There are 2 basic methods for generating nanobodies. One is a display method, and secondly, mass spectrometry-based method, which was really pioneered by Brian Chait, who's on the call here, and his colleague Mike Rout in their labs. In the display methods, generally one wants to inoculate an animal. In our cases, we're talking about llamas as the animal that's immunized. We draw the plasma B cells, and then you can amplify the VHH domains with primers that are common to regions in all VHHs, and you PCR amplify them. And then you can sequence them on the one hand to determine the complexity of the library. And on the other hand, you can also clone them into any vector, appropriate vector for expression and surface display. For example, in this diagram on yeast, but also done in phage or coli. And then once you've got them into these expression vectors and synthesized and produced on the surface of the cell, you then want to go ahead and isolate them. Nowadays, people use magnetic beads with their antigen coated and use those to pull out the cells that are expressing the nanobody of choice. And that can be done by MAX, or even if you've got a fluorescent tag on it, you can isolate those binders by fax. And then you can iterate in this process to enrich for high affinity antibodies or nanobodies if that's your choice. Then to identify them, of course, and there's two basic methods. One, you clone them out, streak them to single colonies, sequence them, and then you've got them expressed in on the surface of your yeast in this example. Or you can just simply PCR amplify all your captured VHH sequences and analyze them by next-gen sequencing, as shown on the bottom there, and you can get a large repertoire of antibodies this way, and you can then order up clones and put them into coli or appropriate expression vectors. The second major way nowadays is to inoculate the llamas, PCR amplify again, and you sequence that library to generate, or you sequence that that RNA to generate a library, and you put that library in a database, and then on the other side, you affinity capture antibodies or nanobodies by typical affinity capture methods, and then identify the VHHs by mass spectrometry and using the library that you generated on the by sequencing as a basis for identifying the amino acid sequences and nucleotide sequences, corresponding nucleotide sequences of the antigens or the antibodies you've captured. And that again can generate a very, very large repertoire of nanobodies. I'm sorry, I should have advanced. That's mass spectrometry shown on the right-hand side there. Then, of course, once you've identified your nanobodies, you have to characterize them. This is just some of the things that we've done to characterize nanobodies against SARS-CoV-2. It doesn't matter whether they're generated by display or mass spectrometry. You assay them for expression and binding to your antigens, do a lot of kinetics and stability analysis to make sure that each of your nanobodies is behaving as you hope. Bind them to epitopes and try to identify where they might be binding. In this case, we were looking at binding to the spike antigen on SARS-CoV-2. Assay them for neutralization or whatever your interest is. In our case, we also test them, the best neutralizers for their ability to neutralize virus and variants of the virus in mice. On the top right is shown spike with a nanobody bound to it on the right-hand side. That's the RBD with a nanobody bound. So these are done by a number of different methods. Cryo-EM would be ideal, but when you're dealing with about 116 nanobodies, cryo-EM becomes impractical, and so often modeling approaches are used. And then you test them in what we found that was really interesting about nanobodies is that if you test them in combination, they can have synergistic activity. So what that means in our case is that we can bind two or more nanobodies to the same spike protein, and they enhance one another's activity. And so in some cases, that enhancement can be over 10,000-fold, which is really quite remarkable, and not a property that one tends to see with IgGs, and that is because IgGs are so much larger, they tend to sterically occlude one another. So this synergy is turning out to be a very common property of nanobodies, and we're quite excited about that. So some potential uses for nanobodies. I've already mentioned some. Of course, research reagents. Use them for affinity purification, imaging, blotting, as you would typical antibodies. Structural biology, they've been used to capture different active conformational states, for example, of G protein coupled receptors. In infectious disease, where part of my interest lies, they can be used in diagnostics, therapeutics, and/or prophylactics, and as Brian mentioned, by inhalation, perhaps, because they're so robust. So you can imagine inhaling a nanobody protecting you against SARS-CoV-2 or RSV. They've been used or explored in immune and inflammatory modulations, for example, in rheumatoid arthritis or psoriasis. targeting these various cytokines. In cancer, for diagnostic imaging, they're fantastic for in vitro and in vivo imaging because of their affinity and penetrance, tissue penetrance. Target delivery of chemotherapeutics is a potential. CAR T therapies, where you express an antibody for targeting CD19, for example, or CD20. But at this point, there's really only one FDA-approved use for nanobodies in humans, and that's for this rare disease. It's a clotting disorder, and the target in that case is von Willebrand factor. And so I'll pass it back to you, Jackie, for questions perhaps, and continue the conversation.
All right. Thank you. Thank you to our speakers for that lovely introduction. We already are getting questions from the viewers, so I'm going to start with some of those questions before we launch into the panel questions. So the first question is, and I'm going to direct this first to John because I think he alluded to this in the portion of the intro. How is the half-life of these nanobodies tuned or controlled?
So that's an interesting question. I think in many cases, a short half-life is beneficial because the kidneys will clear the nanobodies quickly. And so if you're delivering a toxic substance, for example, you want that to be cleared quickly. However, in other cases, you might want them to hang around for a while. You might want to put an FC portion on an antibody. That tends to increase their half-life. Another interesting way is to turn a nanobody into a dimer with one nanobody to your target of interest and a second nanobody targeted against, for example, human albumin. Now that binds to albumin and dramatically increases the half-life in circulation. So there are many different ways. You can even just increase the size of an antibody if that's something you wanted to do as well.
Okay, Natalie, Brian, do you want to add anything to this question?
No, I think John did a good job.
Okay, all right, no problem. Yeah, no, that was fine. Well, then we'll just launch into the panel, the first panel question then, which is, how have VHH antibodies changed the direction of your work and research, and what opportunities have they afforded? And we'll start with you, Natalie.
Sure. So I think, so my company, we partner with other companies to discover VHHs against a variety of targets. And so before VHHs, we were doing conventional antibody discovery, and kind of one thing that has certainly changed with VHHs is the types of targets and the goals of those projects. So, you know, kind of I think this was alluded to by Brian, or even just flat out said by Brian, is that VHHs cover a very different epitope space than conventional antibodies. You know, the fact that they seem to get into little crevices and other concave epitopes that conventional antibodies don't seem to access makes them very useful in our hands as enzyme inhibitors, and then also, you know, kind of different ways to block natural ligands from you know, binding their receptors. So I think the fact that they add a lot in terms of the parts of the target that they bind to compared to conventional antibodies is really what's created opportunity for us.
Okay, thank you, Natalie. Brian, did you also answer the question? How?
Yeah, you know, some of the initial uses that we put nanobodies to are somewhat mundane in that, you know, what we were interested in was inexpensive reagents. You know, we're a research lab and we don't have infinite money. So we do a lot of affinity isolation, a vast amount of that, and a lot of it is against something like GFP, associated with many proteins. If you actually look at the world of proteins, it's been estimated that the community has spent billions of dollars, literally billions of dollars, tagging all kinds of proteins with GFP in animals, in many cell lines, and so on. And so very early on, we decided we wanted to pull out GFP so that not only could we visualize these proteins, but we could find out what they were associating with in cells and so on. And so we started making anti-GFP nanobodies, of which we've made a great many now, and we've shipped them off to hundreds of labs around the world, and we're saving people money, and ourselves as well. And they're very, very good reagents. We can get picomolar affinity. So even a mundane sort of thing like that is quite meaningful. And as John said, we're terribly interested in infectious reagents at the moment for obvious reasons. And we're quite excited about the space, the epitope space that nanobodies seem to access. We're getting better and better at making lots and lots of nanobodies against things like SARS-CoV.
Okay, thank you. John, can you also answer the question of how you have used VHH antibodies in your own work, please?
Sure. Thank you. Yeah, you know, Brian and Chade and Mike Rudd have really established this pipeline for making nanobodies by mass spectrometry. And, you know, Brian and Mike and I have been longtime colleagues for many, many years. But then when the pandemic came, the opportunity was clear, and the responsibility actually was clear. And so in that, you know, we decided to pursue spike as an antigen, but we've also pursued other SARS-CoV-2 proteins as antigens, and we're generating a lot of nanobodies against them. We also intend to, you know, we've explored their use as diagnostics, we've explored their use as therapeutics, as I've mentioned, and I think that as therapeutics, they're tremendously powerful, especially as prophylactics and synergistic combinations. But we intend, you know, I've been just amazed at how fantastically robust and useful these are. And so we've continued to explore them. We intend to build on this in infectious diseases and other applications. For example, in flu and RSV and other very common infectious diseases, especially respiratory infectious diseases, where we think nanobodies have a very clear advantage because of their ability to be used by nebulization, administered by nebulization or inhalation.
Okay, we are getting a lot of viewer questions, so I'm going to pause the panel questions and ask a couple more. So I'm going to stay with you, John, for this question. Are there any known cases of VHH antibodies that can cross the blood-brain barrier to target CNS?
Yeah, that's a great question. There is, you know, I think there is evidence that nanobodies at 15 kilodaltons can cross the blood-brain barrier. And of course, if the blood-brain barrier is compromised, they can cross easier. But I don't believe there's any evidence. I mean, it's very promising, but I don't believe there's evidence to date that they've reached therapeutic potential. That is, they can reach concentrations, or they've reached concentrations that are that provide that therapeutic requirement. However, I think they're extremely promising in this regard, and there's lots of ways that one might think about being able to improve that blood-brain barrier penetrance.
Okay, Natalie, Brian,
Obviously,
would you like to add? Yep, sorry.
No, I agree with John's statement. You know, there's moves to make say dimer nanobodies with one of the nanobodies you know, targeted against receptors, transferrin, that sort of thing, to try to enhance the uptake into the brain. But I'm not aware of too much work in that regard. Yeah.
Natalie, you want to chime in?
This is a little bit outside of my expertise, so I don't have anything to add.
Okay, no worries. Well, then I will ask you the next question. How do the nanobodies differ in clinical trials and response in the body versus monoclonal antibodies?
Well, I actually think that John and Brian probably have a better answer to this. I think that caplacizumab from Ablynx, kind of like paved the way to some extent on how these VHHs behave in the body. And I think that they kind of paved the way in terms of humanization and, that solving the half-life problem. And so I think, now we're seeing a lot more VHH-based antibodies entering clinical trials, and people are exploring all these different delivery modes. But yeah, you should ask John and Brian about that.
Okay, John, Brian, do you want to field that question?
Let me just say that, if I could just say, these are early days for nanobodies or VHHs in clinical trials, and they have been. They've shown, one, Sanofi did a trial, or I guess it was Ablynx before it was purchased by Sanofi, focusing on the F protein on RSV, and they got into, I think they got into phase 2, and then they shut the trial down. It's not clear why they shut the trial down. At least when I look at the data, it doesn't look like there was any safety concerns, but nonetheless, they did. And so I guess it would, I think we're still trying to pave the way, as Natalie said, trying to figure out precisely the safety and the efficacy of nanobodies in human trials. Still early days, I believe. Brian, do you have anything to add to that?
Well, I think it's fair to say these early days, and it perhaps feels a little bit like the early days of monoclonals. They've obviously gone very well, and time will tell. But optimistically, once one finds the right applications, these will become very important.
Okay, we're going to move on to the second panel question. What do you need from VHH antibodies? What qualities would you prefer? Is quality or reliability? We'll start with Brian.
Yeah. Well, the obvious things like high affinity, high specificity. If you're making, if you're trying to target CAR T cells and so on, you better have very high specificity for what you're trying to target. And the potential that I think is quite good is in many of the cases that we've looked at so far, and I guess we've generated probably 300 very high affinity antibodies now, in excess of that anyway. The general feeling is that you can make quite large repertoires of very high quality nanobodies, and for applications I think this is very important, because as you filter it through desirable properties like specificity or high on rate or whatever interests you, many fall away. They will fall away. So having a large repertoire is a great advantage, and I think that's one of the great potentials that I see. John?
If I could just add to that large repertoire comment, I mean, I want to emphasize this synergy thing. I think having that large repertoire allows you to bind nanobodies to different regions of the same protein, and tune or select the attributes you really want, and get that synergistic activity with combining more than 2 or 3 or even more nanobodies together.
Correct.
Natalie?
I guess from my perspective, kind of the needs of the downstream application are variable, and so there isn't necessarily, I think, a golden set of attributes or qualities that I see as being universally useful. We have a lot of projects where people are not necessarily interested in the highest affinity, and there are, kind of like John mentioned, in bispecific antibodies that are binding two different things, and T-cell engagers, you don't necessarily want highest affinity. And so to echo, I think what they've also said is that the fact that we can do immune repertoire sequencing and find lots of different VHHs in this database that have maybe preserved certain properties like specificity or developability characteristics, but you can tune other attributes by mining this immune repertoire, gives us a lot of flexibility, and I guess both phage and the mass spec approach to find the antibodies that have the qualities you want for your particular assay.
Yeah, I guess I'll make a slight additional point to Natalie's great point there. She mentioned tuning affinities, and one of the things that we found is that by fishing out nanobodies with their immunogen, we're able then to elute them under different stringencies and get a very wide range of affinities. So if one does desire medium affinity affairs, one can find that by just eluting with not very stringent reagents. Or if one wants ultra high affinity, one can do that too by eluting and just leaving behind the ones that hang on like crazy. So having this range of affinities tunable is also a marvelous thing to have in one's toolkit. Thanks.
If I could just tie back to that previous question about half-lives. I mean, it's obvious, but I'll make the point anyway. You might want to think about tuning that half-life or tuning the affinity for some host receptor, for example, to in turn tune the half-life of the nanobody within the body. So there's lots of different reasons for thinking about the affinity, as Natalie made that point.
Okay, we're getting a lot of questions from our viewers about humanizing nanoantibodies. How does one do that? I mean, do you work around the framework region, or how exactly? Can any of you cover how VHH antibodies are humanized?
Brian, do you want to?
This has been done primarily by mutating a few key residues in the framework region. I'm not an expert on this, but it's been fairly. We've done it, and to be honest with you, we haven't tested its immunogenicity as a pharmaceutical house would do it, but doing it is fairly straightforward, and one has to make these changes without getting adverse properties, and so far, in the cases that we've tested, this has gone quite well.
Okay, John, Natalie, do either of you want to answer the question as well?
I will just, we've systematically, there's about 6 amino acids in the framework region that we focused on, and systematically changing those. Sometimes changing them has an effect, but we try to maximize those changes to human-like amino acids so that they can be most human-like. But of course, in some cases we can't get all 6. That's all.
We're going to go to the third panel question, which is what advances in VHH antibodies, imaging diagnostics, therapeutics, etc., really excite you or enable your work?
Well,
Natalie, do you want to start off? Oh, I'm sorry.
No,
Brian,
I
go ahead.
was just going to echo John's point. The finding that these synergistic observations, where synergy in the past has been observed a tiny bit with antibodies, but it's quite a rare, and it's not a profound thing to find. What we've been able to show now, looking at a lot of nanobodies against the RBD, the receptor binding domain of SARS-CoV-2, is that it's a feature of nanobodies that we find them very, very often. This great synergy. And I think this bodes well for making mixtures that will be very effective at low dose, will be very potent, and will tend to not prevent escape will elude escape. So I think the prospects are quite good.
If I can go next.
Thank you.
Oh.
Go ahead. No, please go, Natalie. Go, please.
Thanks. The thing that gets me excited about VHH antibodies is their use in therapeutics as multi-specific. So I think the Abling's approved antibody is a multivalent. There's they get kind of increased avidity by having multiple VHH antibodies on the same molecule, but we're seeing lots of different frameworks and structures that incorporate multiple VHHs to different targets, and those could be, I guess, different epitopes on the same target, but just kind of the potential of having these new modalities by using VHHs is really interesting.
So I agree with everything that's been said. I think their use is really only limited by their biophysical properties and our imagination, I would say. There's so much that can be done with them, just like monoclonals have taken off so dramatically. But what I think is important is the potential use in resource-poor settings, where things like monoclonal antibodies are just prohibitively expensive. They can be transported without refrigeration. They can be stockpiled for outbreaks. And there's opportunities for broad efficacy to combat future outbreaks in infectious diseases. So I think it's really, I won't quite go so far as to say it democratizes monoclonal antibodies, but it takes a long way to, improve equity on a global scale and access to these kinds of treatments.
Yeah, the stability. I agree with John. The stability of nanobodies, makes it feasible to be able to keep these materials non-refrigerated for long periods of time. That is a game changer.
Yeah, I should have mentioned it, and I think you did, Brian. But to emphasize it, the cost compared to monoclonal antibodies is really pennies on the dollar. And so that's, I think, an important point.
And to your point, John, the fact is that antibodies, generally need to be injected or given by infusion, and this is a bit of a bottleneck. And so, if one can, at any stage, develop materials that can be put into the airways, as you predict, that would make it, again, a major difference.
Okay, we continue to get even more viewer questions. So a common question that we're getting is, what is the preferred source of nanobodies, an immunized llama or a synthetic library? And if one or all of you could kind of talk about how these libraries are generated, that would be great.
You want to do that, John?
Well, I tried to mention, how they're generated by, in terms of screening and identifying them and cloning them out. In our experience, immunizing animals really takes advantage of the natural evolution of nanobodies within the animal, and in our experience, that is increased, with increased injections, for example, of the spike antigen. We've increased the breadth of nanobodies generated. Which is important when we're thinking about things like future or potentially broadly neutralizing coronavirus antibodies. So our preferred method has been and will likely continue to be immunizing. However, there are great examples where people have gone with relatively naive libraries and generated synthetic nanobodies that way that have worked.
Yeah, I completely agree with John on that. We much prefer inoculating an animal and let it do its job. But even we like our mass spectrometric method for identifying these things directly from blood. But if one does have to use display, I think it can be used quite effectively if it's used in the right kind of way. We have some experience with that now, so it's a fairly subtle story.
Yeah, I guess
Go ahead, Natalie.
Oh, I was going to say, I have to, full disclosure, we're a big subscriber to the mass spec-based approach that Brian and John have been using. And in that case, using an immunized animal is important. And my perspective is that often nature does a better job of engineering certain characteristics than we do, and so we should let nature do kind of as much of that as possible. Of course, my background is computer science, and so I'd love to see more in silico engineering happening. And so I think the future is definitely going to still see synthetic and naive libraries, particularly synthetic libraries, just because all of the obvious reasons why you'd want to do that. It's faster. There's huge heterogeneity across animals when you do immunization. Not everything is immunogenic, so I mean there's always going to be a place for the other libraries as well.
Okay.
That's great. I was going to repeat.
Oh, go ahead.
I was going to say what you said, Jackie, or Natalie. I was going to say what you just said. So that's great. Thank you.
Let's, I guess, dive into the next panel question. What are some of the disadvantages of VHH antibodies over conventional monoclonal antibodies?
Well, we've talked about some of them. Unless you have some mode of keeping them in the body, then rapid clearance can be a major difficulty. And so, especially if you want something to stick around for a long, long time. By their very nature, a bare nanobody does not have an FC effector domain. So if that's important for its utility, either one has to put one on or go to normal monoclonals. And one must admit that there's possible the possibility of immunogenicity needs still to be explored somewhat. So those are the three things I can think of off the top of my head. Any additional, I wonder?
No, I think those are the from my perspective, those are the key points. I mean, the FC. I mean, if you want to do antibody-dependent cell killing, for example, in the therapeutic monoclonals, you simply cannot do it with a nanobody without putting an FC on it or devising some other strategy. And so, and I think again, as Brian mentioned, these are early days, and so we really need to understand, especially as if an antibody is going to be injected, the immunogenicity and.
But Ron, would you perhaps weigh in a little bit on something I know little about, antibody-induced enhancement, one of the downsides of regular antibodies?
Right. So, this is an antibody-dependent enhancement, a very common thing, especially when it comes to dengue infection, in which the FC portion of an antibody is used to bind to a host cell and brings, effectively brings the virus into the host cell and increases infection. And, it's at the beginning of the SARS outbreak, the SARS-CoV-2 outbreak, we were actually concerned about that. We thought that it might be, there was evidence in the literature that SARS-1 had some antibody-dependent enhancement. Luckily, happily, that wasn't the case. Happily, the monoclonal antibodies that we have, that we've used work, and the antibodies we've generated ourselves, naturally work. But this ADE is something that one of the reasons we got into nanobodies in the first place, is sort of hedging against that potential.
Natalie, do you want to chime in? If not, I can ask the next panel question and have you start off.
Yeah, I guess the reason that we use, or the thing that we appreciate about VHHs is much more mundane, I guess, as Brian said. It's just that they're easy to produce, and that, I guess any idiot with some E. coli can make VHHs. And that came off wrong. Very smart scientists can make VHHs and, in high throughput and screen them very quickly.
But I think probably you've hit on quite an interesting point that is of some concern in the pharmaceutical world, is that making antibodies completely the same every time is a difficult task, because there's variability and sort of glycosylation from batch to batch and that sort of thing. And that is certainly something that one doesn't have to worry about with nanobodies. The batch to batch reproducibility is ridiculously high. So that's actually maybe a point that one should keep in mind. Yep.
Okay, I'm going to jump to the last panel question here because I see that the hour is almost upon us. What do you think is going to matter to scientists navigating the life sciences landscape in the next 10 years?
Over to you, John.
happy to start.
Okay.
I think I'm a systems biologist, and I think technologies and computation are really transforming the way we do research. And the complexity of biology is really the challenge for the next generation to try and figure out, to understand, embrace this complexity, and try to address it. And to do that, I think we need team science. We need mathematicians, physicists, chemists, physicians, etc. And I think scientists of the future need to be thinking about, or at least the next 10 years, need to be thinking about how to leverage being part of that multidisciplinary team of scientists as best as possible. And we as academics need to embrace this change. We need to recognize things are changing. And so there are things like authorship, multidisciplinary grants, recruitment for diversity of experience and disciplines are all going to be important things for the future of science, I think, for biological science, I would say. I think the days of one scientist, one protein, one career are limited, if not gone.
Well said, John. I think there's very little that one can say positive about the present pandemic that we're involved with at the moment. But one of the things that has been a bit uplifting has been to see the way that many groups have come together and rather selflessly joined in taking on these hard problems. So, under stress and people can behave very well.
I guess
final thoughts?
Yeah. So that completely resonates, the interdisciplinary nature of things, and I think the kind of like the important part of that is connecting kind of the early basic research with the end state of actually, helping patients or helping health. And so I guess, what I think is actually going to be crucial is faster build, make, test cycles. So whether that's things like good models for patients, getting away from kind of 1970s, models for things like how is this going to behave in the body, how are these drugs going to interact with each other, better ways to screen the things that John and Brian talked about with synergies of different drugs. I think that's really where a lot of focus needs to go, is that we can get to the end point of what all of our research is about faster.
Okay. Unfortunately, we'll have to stop here as we've run out of time. Many, many thanks to all of our panelists for being with us today. It's been a delight talking with you all. I learned so much, and I hope our audience did as well. Please go to the URL now at the bottom of your slide viewer in the Resources tab to learn more about resources related to today's discussion, and look out for more webinars from Science available at science.org/webinars. If you'd like to send us your thoughts for this webinar, please email at webinar@AAAS.org. Thank you once again to our panel and to Jackson Immuno Research for enabling this conversation through their kind sponsorship. Goodbye, everyone.